So I'm really happy to see everyone here today, and I'm really excited to welcome our postdoc invited speaker and first speaker in the Thursday Seminar series for 2023, Dr. Rob Denton. Rob did his Bachelor's of Science degree at Ball State University in Muncie, Indiana. This is where Rob and I met. Rob was looking at being a large animal vet. I was pre-med, and we both decided after we became friends, not necessarily because of that, back with this, we're gonna study nature, and the rest is history. He went on to do a Master's of Science at Eastern Kentucky University, studying the effects of wetland reconstructional, amphibian ecology, and population demography, before going to do his PhD at Ohio State University in Lyle Gibbs' lab, working on the genomics and population genetics of Ambystoma salamanders, like the one you see here. Rob and I were in the same lab during our PhD, and it was so awesome to have sort of back and forth cheerleaders and friends and systems of support. So find your support person and cling on to them. It'll make this whole stressful experience a lot more easy. You want to do a postdoc at University of Connecticut before getting an assistant professor position at University of Minnesota Morris, a PUI up in Minnesota, before moving to his current position at Marian University in Indianapolis. He's back home in Indiana. Rob has published 22 papers in journals like Evolution, Molecular Ecology, Genome Biology and Evolution, and Functional Ecology. And I think one of the reasons that the postdocs elected Rob as our invited speaker was that he is a shining example of successfully integrating a research program with a highly rigorous, structured pedagogy and teaching program at a PUI institution. So Rob currently teaches molecular genetics and bioinformatics at Marian, as well as a professional development course. And he takes these students from his classes, and these students often end up in his lab working on their own semi-independent research projects. He has 2 of his 6-page CV that he sent me dedicated to his 32 undergraduates that he's mentored over his career and all of their achievements and awards and publications. So I think that says a lot about his priorities. But while doing this, he has maintained federal funding for his research. Currently holds a U.S. Fish and Wildlife Services Recovery Challenge grant to help conserve and understand population genetics of frogs in the Sky Islands of the American Southwest, as well as a recently received NSF Career Award to study Ambystomatid salamanders. Not only does he effectively conduct his research and teach, but he also is a triple threat. He has done a heck of a lot of service, and I'll just name a few things here. He's the chair of the Society of the Study of Amphibians and Reptiles Mentorship Committee for students, and founded that program for SSAR. He's the herpetologist. He's a member of the Herpetologist League Diversity and Inclusion Committee, and he is an organizer for a university collaborative effort, Art Plus Biology, which seeks to help make science more accessible at his university through collaborations with local artists. Rob's authored more than 100 blog posts for the Molecular Ecologist blog, and his work has been featured in places like NPR's Science Friday and a story titled Unisexuals, a story of salamanders and sex. And finally, he has recently produced a collaborative, highly produced video and infographic series titled The Secret Sex Lives of Animals, which he says is a project slated to challenge humans' bias toward bisexual reproduction in animals. And with that, and without further delay, we're excited to have you challenge our ideas about how animals reproduce and hear about unisexual salamanders, the genomic, evolutionary, and ecological consequences of strange vertebrate reproduction. Rob.
Wow. Thank you so much, Matt. That was so very. Thank you all for being here. There's no chairs for Kamala Harris to be in this room. I might regret that. I'm honored to be here. This has been an extremely intellectually satisfying day. For me, it's an honor to be invited by the postdocs, of course, and to be voted in this position and share this work with you. I'm going to jump right into it. I've tried to make this as eclectic as possible. Try to include things for everyone. And this story is going to end in a very different spot than it begins. So follow along with me, if you will. And where this starts is when I started my PhD. Of course, like many of you know in the room, you sit down with your PhD advisor and they give you this recommendation to find these big questions in your field, right? And try to drive them forward, spend your chapters driving those questions forward and finding out new things. And I thought I was being really ambitious, so I wanted to go after a big question in evolutionary biology. And that's why is sex so common, right? This is sometimes referred to as the queen of problems in evolutionary biology. And of course, there's this huge, incredible body of literature about this problem and the pros and cons of sex, its costs and benefits. And it's more complicated than it seems, but it boils down to a really simple idea. And most of you probably already have a really firm understanding of this. If you have two species up here on the board, oh, can I get this thing to go away? No? Yeah, more. I'm doing it left-handed. Hide this one. Oh, thank you. We have two species, two populations we're considering here, one that's sexually reproducing and one that's asexual, in a sexually reproducing lineage here. Oh, did I not click on this now? Oh my gosh, okay. Of course, only the females are directly reproducing. Each of these parents is splitting roughly half of their chromosome content over and over compared to an asexually reproducing species. Each individual directly reproduces 100% of that genomic content to the next generation. And we understand some of the economic arguments here. This quickly outruns the sexual species. Why do we reproduce sexually? And these ideas of sexual reproduction in vertebrates and animals are so ingrained that when things contradict those ideas, it's really surprising to everybody, not just scientists, the general public. There's all these stories like this, whether it's a Komodo dragon or a shark or a snake, these virgin births of these females that never come in contact with males and suddenly have a viable clutch of eggs. In reality, this is the tip of the reproductive diversity iceberg, right? There's all these fascinating systems that scientists have worked really hard to understand. From those that are completely clonal, from generation to generation, to those that exist as either male or female and can use reproductive material from other species, to those that are somewhere in between, maybe hybridogenic and semi-sexual, depending on the generation, or those that can change their reproductive anatomy based on social cues during their own lifetime. These systems have produced a lot of good reasons why sex is so common, and a lot of those boil down to the evolutionary advantages of having that genetic diversity. They're present for when things change and adaptation needs to happen. But a lot of these studies are essentially comparing two ends of that dichotomy. Here's a sexual thing, and here's something that is non-sexually reproducing. We know there's a lot of gray zone in between. And that led me to thinking more carefully about this animal on the screen that I think is one of our best examples of that gray zone in between. This is a unisexual Ambystoma salamander. So the genus here is Ambystoma, also known as the mole salamanders. Unisexual, as in it's all female, as defined by its single mitochondrial lineage. One haplotype that's being handed down daughter to daughter to daughter to daughter. At the same time, the nuclear side of this is in multitude. These are polyploids. This is the oldest known vertebrate polyploid, about 5 million years old. Now, these animals reproduce in a really strange way, and I'm going to walk you through what that looks like from a salamander eye view. So, not too expensive. So, this female is going to arrive at a wetland in the early spring. Ponds melt, water fills these vernal wetlands. She is a triploid in this case. So, 3 sets of chromosomes, and circling them here to kind of delineate where they are. This animal proceeds to pick up a packet of sperm produced by a male of one of 4 to 5 other species are possible here. Picks up that spermatophore, it initiates egg production in this female. She lays her eggs, and most of them are clones of mom. The same set of 3 chromosomes in this case. This isn't so weird. There's fish that reproduce this way. It's called gynogenesis. What makes these animals unique is the frequency at which that male contribution makes it into the next generation. Either being tacked on an increase in ploidy, or in some seemingly rare cases, actually substituting in for a set of chromosomes that was there previously. This reproductive mode is really weird. It's got a fun name. It's been dubbed kleptogenesis, right? Name for this stealing of sperm, right? It's a very charismatic idea. You can find these animals all over where we are here, the Great Lakes. So this gray outline here is where we find these unisexuals. They're primarily interacting with the males of two species, Ambystoma laterale, the blue spotted salamander on top, and Ambystoma jeffersoni, the Jefferson salamander. A little bit of nomenclature, because this gets complex really quickly, as you can imagine. We call these unisexual biotypes, ecotypes, genotypes by letters that are associated with the sexual species from whence those genomes came. So for example, if this animal has one copy, one haploid set of chromosomes from Ambystoma laterale, two from Ambystoma jeffersonianum, we're going to call it an LJJ. So I'm going to use these colors and these letters throughout this talk to stay consistent. How do we know this is happening? Well, we've known about this mitochondrial lineage for a long time. There's some really cool work in the early 90s. The nuclear side of this was much more complex to figure out, as you can imagine. Some of the best evidence from this comes from direct genotyping of mothers and their egg masses. Going out in the field, tracking an individual unisexual animal, genotyping every one of its embryos. Sure enough, almost all of them are exactly the same as the mother, until you find one that isn't. This came from a male of that species. Fantastic direct evidence. And one of the first projects we were doing during my PhD was trying to take a population genetics framework to these similar questions. So we went out to these sites in northeastern Ohio, where we knew these unisexuals were present along one sexual species in each of these ponds, Ambystoma jeffersonianum, which is a diploid, as a reminder. We developed a set of microsatellite loci that only amplified in jeffersonianum, so only amplified these J's. So we knew we weren't getting alleles from the L part of the unisexual. We modeled a whole bunch of scenarios of gene flow between these groups using migrate. And sure enough, as you might expect, there's gene flow between ponds for the sexual species, animals dispersing between these wetlands. But there's also a very strong asymmetrical introgression of genes from the sexual species into the unisexuals in the same pond. Not surprising, this arrow is at kleptogenesis happening in the data. What might be surprising is how frequent this analysis suggests that this happens. So if you ask this to quantify, according to the way that migrate does this, which is a little bit tricky to interpret, this number is on order similar to the gene flow between wetlands. Might be a little higher than you would think. This doesn't really tell us what I was initially very interested in. What we want to know. So there's something about that data that captures this kleptogenesis happening, but it's at a really shallow time scale, right? Microsatellites at just these limited set of ponds. This is a broader view of the evolutionary relationships between some of these species. This is based on mitochondrial data. Here are those two primary sperm donors, laterale and jeffersonianum. The unisexuals, from a mitochondrial perspective, are more closely related to other ambystomatid species here. We know that this is happening. Genetic information is going from these lineages into this unisexual one. No doubt about that. But what's happening farther back in the past? I said these are around 5 million years old. Has this process been consistent over 5 million years? Has it gone up and down? Has it changed as ranges have moved around? We wanted to start to get at this idea. And of course, that was going to require much better data than a set of 14 microsatellite loci. So we went out and gathered samples from across Ohio and Michigan, these sites where we had a really good idea of where unisexuals were and where these sexual species were. 50 unisexuals, 28 jeffersonianum at these 3 in Ohio, and 17 Ambystoma laterale at these 2. We used ultra-conserved element sequencing for this, which is really handy to be able to pull out pieces that are conserved between the species and use them to identify where that piece of DNA is coming from. Is it coming from the L or one of the J's within that unisexual thing? We use that data to score these subgenomic SNPs from each of those subgenomes of the polyploid, and then do a similar thing, model a bunch of different scenarios of gene flow and divergence over a much longer time period in this case. Those results look something like this. I'm going to walk you through them on this phylogeny across time. According to that data and the best supported models, first we verified that that data confirmed that these two species evolved from a recent common ancestor around 12 million years ago, which matches data from previous studies and kind of gave us a little bit of a sanity check that we had the power to do this. The ultra-conserved element data shows the first signal of gene flow into this unisexual lineage about 4 million years ago, followed by about 2 million years of nothing. Divergence only, so no gene flow happened. And then it starts back up again, and it's different depending on which species you're looking at. For Ambystoma jeffersonianum, it's a consistent signal of introgression in the past 500,000 years or so. For Ambystoma laterale, two more distinct pulses of introgression at these dates. So we got this data back, we did these results, we redid them. I was convinced that I was very wrong. This can't be right. We defined this system by this kleptogenesis, this theft of genomic information. How is it possible that there are millions of years where that's just not happening? And we redid it, and I thought I'm really wrong. And my advisor was saying, we got to publish this. Maybe you should be right. And so something that was very helpful around this time when we were looking at this data was the publication of this animal's genome. So this is the Amazon molly. They reproduce gynogenetically, right? So very similar to these salamanders in a way. When this genome was published, the authors used that data to suggest that the molly has gone around 160,000 years without sexual reproduction. In our data, it suggests these unisexuals have gone around 2 million years, divergence only, no gene flow. Now, the difference between these species is that they're living evolutionary life in very different lanes, right? So Amazon mollies can reproduce multiple times per year. They're very short-lived. 160,000 years is 500,000 generations for mollies. These animals can live 20 years. They don't reproduce every year. They live life in the evolutionary slow lane. This is about 250,000 generations. So if you don't think about this in terms of time, but it's terms of generation time, maybe that's not so unreasonable that they've gone this amount of time without that injection of new genetic information. Now, I've said a couple scary words for like grad students now, like microsatellites. Like that's old data now, okay? And the reason for that is that there's lots of limitations for studying these animals and making these types of inferences. One is that they have huge genomes. So the haploid genome size here, in this case, is around 28 gigabases. Gigantic. Hard to sequence. They're difficult to identify in the field. In the field, these unisexuals look identical to the sexual species that you find in the same pond. It's hard to find them and get reliable samples from the field. Compounding all this is that at the time, there was very limited computational approaches for polyploid data. Many of these were being developed in polyploid plants, and they were fantastic, but it was really hard to transition them to this type of limited data in a vertebrate system without a reference genome. And so I waited. We had a big pandemic, sort of slowed things down. And through happenstance, a lot of hard work, a lot of work with collaborators, we've expanded this to do it in a much larger degree. And so instead of having those geographically constrained sites in Ohio and Michigan, we now have almost 400 samples from across their range. Ultra-conserved element data, the same. We're repeating many of these analyses thanks to some generous funding from the National Science Foundation. This is my summer, top of my summer to-do list, is to finish this analysis. More to come soon. So we've established that this is an old all-female lineage, that there's some sort of signal of contemporary historical gene flow that's gone up here. It's changed over time. We're not sure why. Molecular biologists in the audience might be interested to think about, okay, well, but then what do these genomes actually do, right? And what I'm getting at is the transcription happening in these subgenomes, in this polyploid, genes to transcripts. A rich body of plant literature has numerous patterns in polyploids in this various way, from things like cotton, where one of those subgenomes is completely silenced, to others that have differential expression amongst the subgenomes depending on tissue or development time. What's happening in these salamanders that are managing these giant genomes from other species? So we went out and generated some RNA-seq data in a very specific way. So one, from a unisexual individual, a single unisexual individual, there was a triploid, 3 copies from 3 different species, the 3 you see here on the screen. Ambystoma laterale, Texanum, tigrinum, and LTTI. RNA-seq data from all of these individuals, are the 4 here on the screen, filtered it, did sort of your standard approach to trying to make a reference group of orthologs from these sexual species. In this case, we ended up with around 3,000 of these. And of course, looking at the reads from the polyploid, we don't know which of those subgenomes that read came from. And so to do this, we're mapping that data onto this reference. So taking all these little dots of RNA, mapping it into these genes, and then trying to make some statistical corrections for mismapping just based on how closely related the different sexual species are to one another. You can imagine this mapping going a few different ways, depending on which hypothesis you like. So one of these could be that there's a sequence of balanced gene expression across a subset of these genes, or all of them in a more global way, where each of these stacks that mapped up are going to be somewhat global. Alternatively, a scenario where there's unequal homolog expression. One or more of these subgenomes is expressing more of that given ortholog than the others, something like this. This was the working hypothesis, specifically because of this animal, Ambystoma laterale. No matter where you find a unisexual salamander, it's always got at least one L, even if it's hundreds of miles from the nearest blue-spotted salamander population. So I thought, oh, that must be some sort of really important developmental connection between that L. It has to be there for this unisexual to survive. And we're going to see that in this gene expression data. We didn't. That's the we didn't. Overall message here is that most of those 3,000 some genes are not differentially expressed. They're balanced across the 3 subgenomes. That's point number 1. The subset of them that are differentially expressed, these 800 some here, were not differentially expressed in the way that we expected. That data looks something like this. So on the y-axis here is the number of sequences. And then for each of these 3,000 genes, we gave them, gold, silver, bronze, highest, middle, lowest level of expression. I'm going to put in a clustered bar chart here with each of these sexual species. Highest, laterale. The winner here is tigrinum, the TI. It had the greatest number when there were differentially expressed genes. It was the winner most of the time. You haven't heard a lot about tigrinum in this talk because it frequently doesn't interact with these unisexuals. It's more of a rare contributor to this system. So the majority of these genes are equally expressed among the genomes. When they're not, it's not the species we thought it would be. More so, if you look at these in clusters of functional pathways and the rank order of their expression, compared to the sexual species, what's happening in that same set of pathways from that species in the unisexual, they're almost identical in their rank order of expression. It's sort of an alluring idea that these genomes pop into this unisexual system and they just keep doing the exact same thing they were doing in the sexual species. They're in a completely different lineage. They're expressing genes in the same rank order. There's very little change here. We've expanded on this. So this was back when transcriptome data was very, very expensive. It's almost like laughable to tell some of the people in this audience about one single animal transcriptome, right? And now we have over 30 from different genotypes. So comparing LJJs to JJs to LLJs. And sure enough, we're seeing almost the exact same pattern. Equal genome expression seems to be the norm across these. All right, maybe you have a behavioral or ecological bent in the audience. And from the very beginning, you thought, what about why do these animals even coexist with one another? Imagine the push-pull in this system, right? So these unisexuals require sperm from a species that they share an environment with. They have similar developmental times. They emerge at the similar time. They eat the same things. These sexual males, it makes sense for them to be able to identify females of their own species instead of these unisexual individuals. There's also a mate choice component to this. How do they manage to not drive one another extinct? There's a really fascinating body of literature about this very thing, about sexual parasitism, and really good modeling work that identifies some of the life history traits that might mitigate these interactions and allow them to coexist without one group driving the other extinct. And I'm not going to labor on these too hard, but I want to focus on 4 of them that often come up and show you what evidence we have for which of these groups is favored in that life history characteristic. The first of which is mate choice. So if these males can differentiate between females of their own species and these unisexuals, it would make it very easy to not waste that sperm output, and it's a disadvantage for the unisexuals. And surely that's the case. There's some evidence that shows that males can figure out this is unisexual, this is a female of my own species, and move and make a choice towards that animal. Advantage, sexual species. Second category here is fecundity. And like many all-female groups of vertebrates, unisexual Ambystoma have relatively low fecundity. One of the hallmarks of ponds that have a lot of these animals is you go out in the spring and you see these egg masses floating in the water, and they're just kind of weird. They're kind of tattered, they're fungal on the embryos. Not as many of them successfully make it out of that life stage. Advantage, sexual species. In organisms like this that have an intermediate life stage, like the larval stage that's aquatic for salamanders, this is a really important time. It's very competitive and sets these animals up for success when they leave these wetlands. Seems to be a wash. They don't seem to have much of an influence on one another, one more competitive than the other when they're in these common experimental situations. And the last life history characteristic, it seems to be important here, is dispersal. And this one is related to organisms that are competitive in this way, these sexual parasites being able to exploit kind of novel habitats that are at the edges of where these sexual species are, and often are better dispersers in the same shared landscape. We don't know much about dispersal in these organisms. If you plug this information that we knew at the time into these models for sexual parasitism, it overwhelmingly predicts that these unisexuals should be extinct. They have no reason to be successful given these disadvantages. So we thought dispersal might be important, and we wanted to test this hypothesis that these unisexuals have greater dispersal ability than a sympatric sexual species. So given the same landscape, these unisexual animals are either going to disperse more often or farther than sexual symmetry. We did this in a couple ways. Out in the field, we isolated these sites in Crawford County, Ohio, typical agricultural area of northwestern Ohio. The reason why is because they've been previously monitored to a really detailed degree. We knew where all the ponds were, what types of animals were in them, and could accurately access them and survey them. This is what they look like from a salamander point of view. They're these beautiful fertile wetlands in these woodlands, usually behind people's homes. You spin around and walk 10 feet, and you're looking at miles and miles of corn and soybean fields. A very intimidating landscape for a salamander. Advantageous in this case because it makes it easy to find animals that have dispersed because they're isolated from one another to a pretty large degree. So we surveyed 15 of these wetlands for 3 years. Ended up taking tissues from around 150 unisexuals, around 300 of the local sexual species, Ambystoma taxanum. We used those same species-specific microsatellite loci, since we were genotyping hundreds of these animals, in an assumption-free network, the DAPC, discriminant analysis principal components, to try to identify animals from a given site that stuck out like sore thumbs in the data. We could assign them instead to the likely site where they were born, 3 miles down the road. We're calling those animals dispersers when they met a certain threshold. So an individual assigned to a site other than where it was sampled with a high posterior probability in these analyses. Here's what it looked like. We found some. The number of dispersers, relatively similar in this data set, and relatively low, as you might expect, and would fit a lot of the other ambystomatids. But if you looked at each of these individually and calculated up the distance, the minimum distance they would have to travel from where we assigned them, their place of birth, to where we found them, where they had dispersed to, that distance is very different. It's much higher for the sexual species. These numbers are a little fuzzy. I think it's more helpful to show you what this looks like on a map. These are the sites on Crawford County. On the top is Ambystoma taxanum. On the bottom are the sites where we sampled the unisexuals. I'm going to draw in arrows here that represent a dispersed animal. Now, of course, they didn't travel in a straight line. Like, these are much more complex than we're showing here. I just want to give you an impact of what this looks like. So these are the dispersal movements of Ambystoma taxanum, hallmarked by these long distance moves, especially over this middle part of the map. There's a reason there's no dots there. It's almost all farm field. There's very little wetland habitat in between. Compare and contrast that to the unisexuals, mostly adjacent hops between ponds. A pretty stark contrast in this case. Also, the opposite of the prediction that we made. It's a real theme. I gotta go with that. So instead, we have similar percentages of dispersers and a much greater dispersal distance of Ambystoma taxanum instead. All right, so we had lab meetings to talk about this data. Oh, it's really interesting. Let's talk about all these complicated evolutionary reasons why we might see this pattern. And then someone in this lab meeting sort of jokingly proposed, what if they can just walk further? Yeah, what if they just walk? Oh, wait, what if they can just walk farther? Like, think about this scenario. They have a limited amount of time in the spring to move before it gets too warm or the rain goes away. Animals that have the ability to walk farther in a given time are more likely to locate one of these other reproductive habitats. Can they walk further? In other words, do they have higher locomotor endurance? We compared sexuals to unisexuals. Okay. Went back out to the field, got some adult animals, brought them back to the lab. We borrowed a salamander-sized treadmill. The tread is made of wetsuit material, so you can keep it wet. It has a motor, so you can dial in the speed. You ask the salamander nicely to walk on the treadmill. These are not sprinting animals, right? So once they're convinced that you see them and that you're potentially could hurt them, they're walking at a steady speed. They're not gonna sprint. Now, this work it's gonna look really cute when I show it to you. And I want you to know that it is very charismatic. It is also awful. So we did this in a cold chamber at like environmentally, relative temperatures to what was going on outside in the freezing spring of Ohio. The reason why it was so miserable ended up being because some of these animals walk on this treadmill for an awful long time, okay? So keep that in mind when you watch this. But this is what it actually looks like in practice. So let an animal walk. After every 3 minutes, we took it off of the treadmill, tested it for writing response. Can it write itself, or is it too exhausted to be able to write itself? If it flipped back over, spray down with some water, put it back on the treadmill, another 3 minutes. Over, and over, and over again. Now, The reason why I did this over and over again is because some of these animals could walk a tremendous distance on these treadmills. And the one that could walk the farthest distance were the sexual species, Ambystoma texanum. They walked around 4.5 times farther than the unisexuals. So about 160 meters on average compared to about 35 meters. Now, these numbers don't mean a lot to you, but if we scaled up body size from a salamander to a normal human size, this is like you going on an 80-mile jog before you're like just too tired. It's a huge jog, right? It seems crazy. And a stubby little animal on this treadmill. This is what the data actually look like. So on the y-axis here is the log distance traveled. Here are the two groups, texanum and the unisexuals. Each of these circles is an individual's trial value, and the gray bars are the means here in this. Now, one conclusion that could be made about this is that Ambystoma texanum are just these distance marathon runners. They have some amazing ability compared to all other Ambystoma. We were lacking some comparative power in this regard. We happen to have individuals from these other species in the labs. We thought, well, let's try them and see what those numbers look like too. And sure enough, they're on the same level as Ambystoma texanum. So we clearly see, a disability in locomotor endurance for these unisexuals. This is that data that I just showed you on the right. This is the same type of figure made using the genetic information, the dispersal inference we made with the genetic information. This was a really fun project because it allowed us to think a lot about a pattern in genetic data matching the functional thing that we think would produce that pattern, which is a really satisfying thing to do. Again, this didn't really answer the question. If dispersal doesn't favor these unisexuals, well, then what does? That's another strike against them in the models that predicts coexistence. But the real legacy of this work, besides that video, I guess, is that it made us think a lot more about the physiological limitations of this system, and potentially some of the penalties that these polyploids are paying for this complicated genomic scenario. In some studies that we've done and others have done, the unisexuals are terrible compared to the sexual species, like locomotor endurance. Other things that we've done, like tail regeneration, an undergraduate did this study with Matt and I at Ohio State, and showed that these unisexual polyploids could regenerate tail tissue around 1.5 times faster than the sexual diploids. What's going on here? Well, it made us think a lot about the genomic basis for these types of pathways and activities, things that are metabolic in nature and require exertion and cooperation between mitochondrial and nuclear genomes, versus those that might benefit from the raw horsepower of expressing more genes on more chromosomes. What makes this interesting in this system is that these unisexual populations contain a set of nuclear chromosomes that are both geographically and temporally isolated from their nearest source. So I'll show you this again. Keep in mind that some of these animals in Kentucky, there's a lateral genome down here that's 300 miles away from the nearest blue-spotted salamander habitat. It's isolated. The other component of their nuclear genome is just coming in all the time from the sexual species that they're interacting with. In other words, each of these unisexual polyploids is a combination of old and new, an old divergent mitochondrial haplotype and this rotating cast of nuclear chromosomes. Gathered some preliminary data on this idea, talked to collaborators, thought a lot about how we test some of these things, and we were just overwhelmingly fortunate to be awarded this NSF career grant to ask this question about mitonuclear mismatch or conflict in these animals. How do they manage this mismatch or this predicted mismatch? Let me explain a little further. So in other clonal or asexual groups or sexual species, there's clearly been identified this pattern of maternal bias in genes coming from the nucleus that need to interact with the mitochondria, mitonuclear proteins. A preference for those that have some level of coevolution between mitochondrial and nuclear genomes. In asexual species, where those mitochondrial haplotypes acquire mutations because they can't purge them, they're clonal, there's records of that causing inefficiencies in the processes that are happening in the mitochondria. These unisexuals offer the weirdest possible combination of these ideas. Because their mitochondrial haplotype didn't co-evolve with these nuclear genomes, it's separate and independent. It has its own evolutionary history. These nuclear chromosomes are shifting in and out of that lineage over time. Some might be newer or older than others. Are those older ones the ones that are contributing to these interactions and the newer ones causing mismatch? We don't know how they manage this, but we're going to find out. So we're doing metabolic experiments, we're doing mitochondrial function experiments, using whole animals combined with a lot of genomic stuff. Never been more excited to do this work. So fun, so fortunate. These career awards, of course, involve the authentic integration of this research with education. It's a huge part of it. And one of my favorite parts about being at a place like Marian, a primary undergraduate institution. And so it's also enabled us to do a lot of fun things combining research and teaching. That includes a fantastic collaboration between our department and individuals in art and design to force, or not force, to let our students between these courses work together and find out how their products and how their processes, how they do their work can illuminate one another. So we're starting this next year, pairing my biostats class with a drawing class to see how they work together. It also provides these really direct outlets into classrooms, taking this data, having it go directly into my bioinformatics class. Let's assemble these mitochondria. Let's try to identify mutations. Let's make some trees. Fresh out of the sequencer, off my computer, into my classroom. It's also a fun opportunity to provide some of the training that I wish I would have had as a postdoc for my current position. I knew how to do research. I had a lot of training and teaching. I was involved in a lot of service activities. I never had any practice doing all those things at the same time. And so we have a training program for postdocs that we're starting that gives them more of an authentic idea of what it's like to work at a PUI, and it's just been fantastic so far. So many people to thank for this work. I couldn't just possibly fit them all in one slide. From those who funded it to those who work in the field, people all over the country who have helped gathering samples and have enjoyed working on this project and contributing. Before I leave though, I have something a little strange to show you. So it's gonna take a little bit of explanation. It's something strange so far. Okay, the story goes like this. That tissue regeneration paper that I mentioned, led by an undergrad. We were excited, it got published, got into a good journal. The university wrote a little press release about it. We were thrilled, so fantastic. That press release got picked up into some national outlets. Someone from the Washington Post wrote a really nice little article about it. This undergraduate was over the moon, so excited, we were proud. It came out in the Washington Post. A group of us were reading it in my office, and at the time, the Washington Post had anonymous comments section at the bottom of the article. You can see where I'm going on this. And we realized this and thought, oh my goodness, like, what is the average anonymous person on the internet going to say about the study? It had some sort of spectacular title like all female salamanders don't need no man kind of thing. And so, we kind of got it, we steeled ourselves, got up a little bravery, scrolled down to the bottom. And I'm not exaggerating, this is a true story. The very first comment under this article, someone anonymous on the internet wrote us a poem about these animals. Okay, screenshotted it. I've saved it for like dark and depressing times during my career. And I review it, and I always show people at the end of these talks. It's just delightful. So I'm going to put it on the screen, give you some time to look at it, and then hopefully have time to answer some of your questions. Thank you so much. We have time for some questions. We good?
I think so.
Okay, great.
Question?
Randy, go
I
ahead.
got it. So, you did a really good job of addressing all the different disciplines in the room except for one.
Oh, no.
So, from a taxonomic perspective, the entire time you're giving the talks, I'm like, unisexuals. What does that mean? Is that a name? Is that a behavioral group? Is this a, some chunk of? Are they all literale and they do some weird behavior? So anyway, I just wanted. How do you know that they're not all just literale that are hybrids in some places and just using in time of need reproductive strategy?
Right. Okay. Yes. Good question. So the idea here is about taxonomically, what are we talking about here on the species, gradient? Well, context, for a long time, this group was recognized as two semi-independent hybridizations of Laterale and Jeffersonian. They were called Tremblay and Platinae. Until some of that mitochondrial sequencing revealed that they are monophyletic mitochondrially and very different from all the other Ambystomatids. So from a mitochondrial perspective, if I just showed you that tree, anyone in here would say, that's a species. It's as different as any of these other things that we call species. From a nuclear perspective, it makes no sense. They're just this rumble of different nuclear genomes. And so there's somewhere in between where the name is not really appropriate. So I think there's been some discussions about, should they have a name just to be useful, like so it helps us talk about them? And in some states, they still do give them a name so they can give them conservation protection. Things have to have a name to protect them. Broadly, though, we stick with the term unisexual lineage for the most part to describe it. It's a good question.
With your performance data, did you overlay that with the number of chromosome sets in your lineage to see if, like, maybe as the number of chromosomes increases, performance decreases, or inverse of that?
You're talking about the locomotor stuff?
Yeah.
The 98% of the individuals in the study were all triploid. So we didn't have enough. I would love, that's a great idea, and I would love to do that. Most of these populations where you find them, they're all very similar to one another. So this pond, it's all triploid, they're all LJJ. This pond 10 miles down the road, they're all tetraploid, LJJJ. There's places in Michigan where they're diploid, they're LJ, and that's the only kind of unisexuality you find. There's clearly some population-specific dynamics that are happening, and they likely turn over time, where they build in ploidy and then crash back down. And Jim Bogart and others have ideas for how that happens, but there's a lot of work to be done. Am I responsible for calling? Okay.
Yeah, so two questions. One of the, so what percentage of offspring in like a cluster or whatever are clonal versus sort of what's the expectation for that? And also in the performance data, did you see any bias between male salamanders and female salamanders for the sex species?
Yes, excellent. Both excellent questions. So, the second one, is there any bias in dispersal between males and females? No, slightly different, but it's too messy. There's too much variance to say it's different between males and females in this case.
In the performance data?
In the performance data.
Oh, okay.
Yes. And then what was the first one again?
What percentage?
Yeah. So what percentage of offspring in a given clutch of eggs are clonal versus those that might incorporate a new set of chromosomes? It varies, again, quite a bit. Probably in the high 90s, like 96, 97% of them are clonal. There's also some environmental corollaries there. So temperature influence, the higher the temperature, the more likely it is they're going to pop another set of chromosomes into those offspring. So they demonstrated that in the lab and suggested that this is one of the reasons why certain sites have more triploids, tetraploids, potentially pentaploids than others, and also that has been related to climate change, shifting distributions, and those things, of course. Good question.
So I know this is probably speculation, but I was wondering if you have a preferred mechanism you think might explain your 2 million years with no sex, like, or at least no gene flow that you can pick up. Do you think they were literally living sympatrically and they were the unisexual lineage was using a different spermatophore from some other species but couldn't incorporate the nuclear information, or something changed where they're still picking up laterally, sperm packets, but are not incorporating them for some reason, and it changed?
Yeah.
Or something else entirely.
Yeah, so speculations about what could cause 2 million years without this introgression. There's a few of them. So one, some folks in New England have proposed the documentation of egg production without sperm picking up sperm. So maybe potentially there's a way, a mechanism they can be purely cloned. I'm not so sure about that. Likely what I think we're seeing here is we have a limited geographic sampling, and it conveniently is in a place where there's a lot of variation in how glaciation affected where ranges went. And I wonder if we're not just picking up in a very specific area, that time period, out between the unisexuals and one or both of the sexual species. We should be able to figure that out with the better. Oh, yes. Wait.
Well, you showed this one slide, I think it was of the laterally complex and the sort of contributors of the genetic 50% talked about this abundance of tigrine. But then you can talk about it. Where does all that tigrine?
Oh, good question. So they can steal sperm from tigrinum. It's relatively rare compared to the other sexual species. In fact, one of the things that's not so surprising about the data is that if you find an individual that has a tigrinum genome, you know before you know it's in, because they're about 30% bigger, big, beautiful golden eyes. They have phenotypic tells that are associated with tigrinum.
So this is farther south in Ohio, not in the states.
You can find the tigrinum introgression in Ohio, in Illinois. We were looking at photos of them earlier. In multiple places across the range. Tigrinum's distribution, of course, tends to be a little bit more spotty. They're like locally.
Because here, the tigrine actually.
Oh, is that so? Yeah, and yeah, it could have some breeding overlap there. That's cool.
Do they do meiosis at all?
What was that?
Do they do meiosis?
Yes. Yeah. So pre-meiotic doubling is what's causing the ploidy increase.
So if they are triploid, how does the meiosis work?
Yes. Okay. So, okay. So they're matching homologous chromosomes, but there's almost no recombination.
So they have 3 sets. So say you have like L, T, T, I, 3 sets. So 3 chromosomes, 3 copies. How do they meiosis?
Yeah. So doubling prior to meiosis. So LL, those separate. TT, TI, TI.
But normally you have 2 sets of chromosomes. After meiosis, you only get 1 set.
Right.
You have 3 sets. So I wonder how the meiosis works.
Yeah.
Typically, this would cause unbalanced gametes. But in those animals, I don't know what will happen after meiosis.
Yeah, so the cytogenetic studies that have been done show that there's pre-meiotic doubling prior to meiosis.
So after meiosis, they still have 3 sets.
Yeah.
But then they also steal sperm from another species, and they will get 4 sets.
In some of the individuals, yeah.
Then this will increase the sets of chromosomes working.
So it doubles first, then it reduces.
Doubles.
Yeah.
I want to talk to you more about it.
That's a good time to segue. So we're going to do a reception with snacks and beverages up in their kitchen area on the 2nd floor. If folks want to catch up with Rob more in the biodiversity neighborhood area, 2nd floor.
Thank you.
Sorry.